G'day, my name's Mark Hutchinson. I'm a professor in medicine at the University of Adelaide and I'm proud to be part of the founding executive of the Animal Welfare Collaborative. Today I'll be talking to you about pain, some of the fundamental developments in the neurobiology of pain, but then I hope to also expand in some other talks around the importance of our knowledge of human pain and its relevance for sheep pain and that in livestock production, and then how we can advance further the translation of fundamental science knowledge from the laboratory to have real-world impact out in the field in livestock or in conservation preservation of animal species. But today, let me start by talking to you about the ability to translate knowledge to practice. And specifically focusing on this concept of bench to boardroom science and some of our experience in and around this space with regards to pain. I'd like to start by acknowledging country, acknowledging the enduring connection that our Indigenous brothers and sisters have to country, and pay my respect to elders past and present. I have a range of commercial roles. I'm not selling you anything other than the science today, but please do take into account that I am both interesting and therefore conflicted. I'd like to acknowledge the funding sources for the work that's being the work that is being presented today, and then in the other presentations, funded by the Australian Research Council, the Australian Department of Defence. The team is supported by the Sheep Industry Fund in South Australia, Meat and Livestock Australia, as well as the Davies Livestock Research Centre, and through collaborations within the Centre for Nanoscale Biophotonics, which I am the director of. And I must acknowledge that I'm not doing much of the work these days. Rather, it is a brilliant team of collaborators, that being the 200 scientists that are part of the Centre for Nanoscale Biophotonics, spread across the country, the team on North Terrace campus at the University of Adelaide within the Neuroimmunopharmacology group, and then a broader array of collaborators within the livestock sector, for example, Professor Wayne Pitchford, the director of the Davies Livestock Research Centre, through to other key collaborators in psychology and statistics, Mel Humphries and Carolyn Semler, and then the commercial team who actually contracts things. This is not only a science deal, it is also a legal and structure. So Greg and Nadim enable much of the work to occur, funded and in a timely fashion. So what are the objectives that I hope to get across in this first presentation? First of all, I want to really articulate how truly complicated pain is and how much of a frontier science it is. Critically, there is the need to address this complexity with a new approach to the scientific endeavour that is being undertaken to address this challenge, and this is embodied within the field of convergent science. And what I want to paint for you is a picture of how we can capitalise upon the opportunities that are present today to create a non-linear, rapid acceleration path forward, but with the challenges that we're faced to actually achieve this. And I'll focus in this talk specifically on the challenges in human pain, which are directly relevant to the challenges we face in the management and mitigation of animal pain. So why am I talking to you today? Why do I have a research laboratory doing what it does? And I want to start by talking about the global burden of disease in humans. This figure here on the right is a two-dimensional graph of the area proportional to the years lived with disability, broken down into each of the causes of the years lived with disability in the human population. So the larger the area, the greater the number of years lived with disability. Now, this is not usually the figure that gets shown that attracts as much attention. Usually we look at the diseases, pathologies, the things that lead to death. These are not the things that will kill you. These are the things that you live chronically with, that society lives chronically with, that contributes to disability. Now, what you'll notice is that there are some very large areas covered by things that we know quite well. low back pain, musculoskeletal pain, neck pain, depression, anxiety, headaches, drugs of dependence and drug misuse. We also have some small but critical areas of early childhood development, and then quite a large number of other not diseases but events that can occur through life that lead to years lived with disability. But what may not be immediately clear from this graph with all of these different pathologies and disease states grouped is the prevalence of pain across the years lived with disability. So if we project onto this, the diseases that are associated with or directly caused by pain, more than 3/4 of the years lived with disability are diseases associated with pain. Now, this represents a fundamental failure of the Western medicine approach to management of, understanding of, and clearly then the mitigation of pain in humans. And this remains the greatest burden on the health system today, is the unmitigated pain caused by or directly resulting from this broad range of pathologies. Now, why do we still have this? Why has Western medicine failed in addressing this? Why is it that we have not got a scientific solution for this? And at the heart of this, it is because pain is one of the most complicated, insidious challenges that we have, and truly represents a fundamental science challenge today. We can't limit this to just being a problem of one system. And as Prof. Paul Rowland calls it, we need to start reframing our understanding of acute, hyperacute, through to chronic pain, truly as the cancer of the soul, because of its complexity and challenges it presents to all of life challenges. Now, what do we understand about pain? Are we starting from scratch? Well, no, we do understand now that pain is complicated, and one of the best ways we consider and conceptualize pain is that it is a biopsychosocial complex presentation. Now, what does that mean? It means that there is a biological component that we can understand at the molecular level, which is occurring on the scale of nanometers. It means that then we are then connecting to that psychological level at the level of the brain and coordination of events that are occurring over centimetre ranges across brain networks. But critically, when we start examining the social elements of pain, this then means that we're dealing with events that are occurring both inside our body as a result of our interaction with the environment, but also outside the body and the actions of others and their contribution to our actual biology through their interactions with us at that social level. So whilst biopsychosocial seems to just roll off the tongue as a simple way to explain an extremely complex thing, we are dealing with events that range from the nanometer up to the meter resolution. Now, I hope you can also then see that when we start considering the timescales that we're actually operating over, we're dealing with events that are occurring on the microsecond down to even nanosecond scale level, right through to events that are accumulated across years. This is a fundamental challenge of our ability to conceptualize events that actually occur at this complex level, let alone being able to measure it. So this means that with biopsychosocial pain scales, we are dealing with vast time and length scales from molecules to systems levels events. And to be able to then explain and potentially mitigate against this, we must gain insights along the entire time and length scales associated with pain. And in the case of humans, we can start talking to people about their pain experience, but in animals, we do not share that ability on the language scale to actually have that direct communication. Hence, the complexity and communication barriers have led to this persistence of a lack of understanding of the true complexity and what we call the molecular etiology or the origins of what truly pain is. But we know it is complicated. So if we break pain down away from the complexities of just the biopsychosocial state, if we now just speak about, let's call it the wiring diagrams, just the communication of an action potential in the neuronal system, can we create a simplistic view of how simple pain is? And the way I'd like to approach this is to ask the question, is this simply a light switch, pain's on and off? Or are we dealing with a biological volume control approach? So that is, are we dealing with an analog or a digital system? Now, this may appear to be playing a little bit of semantics, but it's critical when we start to even try and break down, when we make a measurement, what are we actually measuring? So let me take the example of the analog biological response. Here we're dealing with what I would more term the biological dimmer switch, where a stimulus is applied to biology, and as the stimulus gets greater and greater, all of the biology turns up the dimmer switch proportional to the stimulus, meaning that all of the biology is responding. So if we looked at the fraction of the cells that are responding, well, as soon as we turn a stimulus on, everybody's responding, but it's the amount of response of each of the cells that is proportional to the stimulus. Now that's the analog response. But if we ask the question from a digital response, more of that binary, let's turn the signal on and off at a switch, here we can have increasing amounts of stimulus applied, and as the stimulus increases, then more and more of the cell population respond. So in contrast to the analog stimulus, where the stimulus was proportional to the response by all of the cells, here we actually have only a fraction of the cells responding proportional to the stimulus. But when we then look at the amount that the cells have responded, or the amount of the biology that has responded, actually it's an all or nothing state. So when there's a response, boom, it's all on. When we then come to make these measurements, and if you're in the biology sphere, you'd be familiar with the Western blot, which is a protein assessment of expression. There are a variety of other assays that you can make a measurement like a Western blot. And I guess I would draw the correlation here to the unfortunately familiar rat test. When you look at those lines on the rat test, this is very much like a simple version of a complex Western blot. The greater the intensity of this line on the rat is actually proportional to the prevalence of the viral antigen that has been either scooped up off of your nose or in your saliva that's gone into the rat test. The key part about this kind of biological response that I'm speaking to here, independent of whether it is an analog or digital response, we see exactly the same response on the Western blot. That is, that even though there are fundamentally different biological processes underpinning this, our tools are unable to delineate an analog or digital response. Now, I guess if we actually apply that to the rat test, is it that there is sufficient antigen coming from all of the biology that you've assessed, and when you're not so crook, you've got all of the cells not expressing very much, but when you are really crook with COVID, you've got all of the cells expressing a lot, so therefore you've got a greater signal? Or is it in fact, when you're not so sick, you haven't got many cells infected with COVID, and therefore there's not very much antigen on the rat test? But in contrast, when we then look at when you are very sick, you have a lot more of those cells infected, i.e. digital switch on, and therefore your signal is higher. I hope you can see that then when we come to actually intervene, if it is this kind of a treatment issue, we've got a lot of cells to target to actually have a treatment effect. If it's this difference, then perhaps if we got in early enough of your biology is not intervened. Now, this is a critical question when we start to think about pain, because when we start to think about pain and nociception, is it operating in an analog sense? That is, when we feel more pain, we've recruited more of the cells to a greater extent, or is it digital? When we feel more pain, more of the cells are recruited. So we've asked this question, and we've asked this question in rodent models by creating a form of sciatic nerve injury by injuring the sciatic nerve in rats, and by changing the total injury extent, that is by the type of ligatures we apply to the sciatic nerve, we're actually able to create a graded response, a graded mechanical allodynia, or that is what was once touch becomes pain, and the grading of that is very indicative of how severe we believe the pain is. The correlate to this would be if you were to be asked by your doctor how much pain are you in from a scale of nothing to 10. Here we would be representing the 10 response, and here in the middle we're sort of asking the question of the 4s, 5s and 6s. Now, what we did in this particular study is we asked the question of what changes proportional to the amount of pain that the animals were experiencing. And we asked this question of their messenger RNA within their spinal column, and we then just plotted this proportional to the amount of pain behaviours the animals were experiencing. And what we got was a lovely normal distribution, nothing special there, but the key thing that blew us away was in these tails out here and here, where we are seeing high negative correlation and high positive correlation to the response. This was showing us that there were 600 genes that were changing their activity proportional to the pain behaviours the animals were in. Now, this 600 genes suggests that that's 600 molecular targets that are changing their activity proportional to the amount of pain the animals are in. Therefore, we're starting to see an event that's occurring in a potentially analog fashion. But we wanted to take this further and wanted to actually ask the question of how many cells are being recruited, asking this digital question. And so to do this, we actually created optical windows, literally visual windows into the spinal cord of mice that had a special protein expressed that allowed their cells to fluoresce, this green colour, just within the immune cells of the spinal cord. And we were able to ask the question over time and proportional to the injury, as to how many of these cells responded proportional to the amount of pain behaviours the animals were in. And what we saw was that the changes were both in the density, the form and the velocity of the number of these cells. And there was a recruitment of these cells. So not only are we seeing nociception being linked to an analog response, but we're also seeing it potentially being a digital response as well. So we want to take this to the next level with some new technologies. And this is where we were starting to look at just the native colour of pain within the level of the spinal cord. Now that may appear to be a strange question to ask because a spinal cord should just look the same colour no matter what's happening to it. But we can actually use a new technology called hyperspectral imaging. Now what hyperspectral imaging does is it shines a range of different colours onto the spinal column. That range of colours, just like if you shine red light with your pulse oximeter, you get a red colour through your finger, which is proportional to your oxygen saturation in your hemoglobin. If we shine other colours into the spinal cord, there are other fluorophores or endogenous substances, proteins, chemicals, other things within the spinal cord that also have photo-emitting or photo-absorbing qualities. And if we are able to then create that information through basically shining the full rainbow of colours onto the spinal column, we get information back as to the specific colours that might be generated by, and I'll call the term the fingerprint, mixing my metaphors here of spinal cord fingerprints, but we are seeing how much is changing within the spinal column and if this can actually be seen on the colour measurement. Now, to give you an idea of the kinds of amazing things we can see, here is some cell culture under a microscope, and this is the hyperspectral imaging and the colours we're projecting back onto this. These are all daughter cells, so they should be the identical cells. But what we are seeing here is that even adjacent cells, which have only recently divided from these other sites, have profoundly different colour or biological materials within them that have the capacity to give us different information. And we can start to see that even within cells, there is a differentiation of what is at the nucleus, what's in the cytosol, and what's at the membrane. Now, we've known that this has been the case, but we haven't necessarily utilised just the native colour of the biology to tell us this, nor have we actually been able to have the opportunity to understand in a dish like this, are these cells responding in an analog fashion? And so I'd argue here the greens might be an analog, but here we clearly have a digital capacity of a different subset of cells to change response. So are we actually seeing both analog and digital capacities of biology actually at operation? So we asked this question of pain, and we asked this question of the spinal column under hyperspectral imaging. And what we were able to do was then ask the question of machine learning of the hyperspectral imaging of the spinal cords associated with animals that had been behaving, what colours are changing proportional to the pain behaviours that these animals are expressing, and then, please computer, show me where these pixels are that are changing proportional to the pain that the animals are in. And oh, by the way, computer, can you please only give me those pixels that seem to clump together in greater significance, allowing us to create this pain mask. Now, what this shows is the areas, I shouldn't say cells, the areas of the spinal cord images where we saw pixels that were changing their colour, in inverted commas, proportional to the amount of pain behaviours the animals are in. And what was absolutely extraordinary to see here was that we saw cellular sites where there are nuclei here, so we know that this is a cell. But critically, we also saw non-cellularized regions responding to proportional to the amount of pain the animals were in. This suggests to us for the first time that not only are the cells responsible for the degree of pain the animals are in, and we sort of knew that, but secondly, that there are extracellular, non-cellular spaces which also appear to be critical for the presentation of pain. The other part to this is that we now know that this is both a neuronal and glial signature based on the types of nuclei we can see, and there is an element of binary nature, that is a digital nature of this, because only a certain subpopulation of cells are actually responding proportional to the amount of pain that the animals are in. Therefore, we are seeing for the first time a digital and analog combined response, creating even greater complexity in our understanding of pain. So, I haven't actually simplified pain at all for you by just taking it down to the simplest of forms, that being nociception. But I do want to now try and explain to you how complicated pain is in these systems by actually addressing how many molecular signaling pathways are actually involved in this complexity of pain when we are dealing with it in its simplest form. So I want to highlight here the work of Ted Price from UT Dallas in the US, who has done some brilliant work focusing in at events that are occurring at the 0.1 to 10 nanometer resolution, and that is specifically looking at the relationships between proteins that are released and the receptors that they activate. Ted's work is only focused on these protein targets. It's only focused on one particular anatomical compartment of the body called the dorsal root ganglia, which is sort of this really important key junction box at the level of our spinal column. And often when people have a bad back and they talk about the nerve being pinched, it's actually the nerves immediately at or in the dorsal root ganglia that is actually being impinged. So it's a relevant thing for human models. Ted's work has really focused on, do you have pain or not? And he's been so simplistic in his approach, which is why I want to focus on this. It's just on the glial to neuron, the immune to neuronal communications. So at each of these points, he is being as reductionist as possible to ask as specific a question as possible. Now, what Ted's shown is that, and I do want to just emphasize, just so that you understand where we are at, we're just looking at the dorsal root ganglia. We're not dealing with the complexity of the spinal column. Really, we're looking just at this simple junction box of information that's contained up here in the dorsal root ganglia. Now, when I spoke about the receptor-ligand pairs, this is important to understand because we are dealing with extremely specific relationships on a cell. So here we have a cell. These big things that are sticking up here are proteins. These proteins can then be interacted by other complex proteins that are called ligands, and so we end up with this receptor-ligand pair. Now, Ted's work specifically focused on neurons, the wiring, talking to the immune cells, or the immune cells talking back to the wiring. The other part that Ted was exploring was this key relationship between these two cell populations at the glial neuronal interactions just within the dorsal root ganglia. So what has Ted done? What he's done is he's looked at the RNA within the dorsal root ganglia and he's asked the question, are there pairs of information between the immune cells and the neurons within these dorsal root ganglia such that the expression of these proteins, and these proteins can either be a ligand or a receptor, are actually matched together? So that is, can neurons release something and tell the immune cells what to do, or can the immune cells release something and tell the neurons what to do? And by doing this in a systematic fashion, Ted's shown that the neurons are able to talk by about 133 different pathways to the glia, and critically, he has linked this to just the messages that go to neurons that are responsible for the sensory information within the dorsal root ganglia. So just those neurons that are communicating the potential presence of a painful signature. So neurons are saying about 133 things at any one time to glia. Glial messages talking to neurons, again, that are just the nociceptive, the potential pain signaling messages, are saying a little bit more than neurons. And for me as a glial scientist, I'm quite excited by this because it says that glia have more to say to neurons in a potential pain state, with roughly 199, nearly 200 signaling pairs identified across this immune to pain sensitive, pain responding wiring of the dorsal root ganglia. Now, why am I harping on about this? Because before I talked about, we saw 600 genes changing proportional to the amount of pain that animals were in, and those were a wide variety of things. We hadn't asked the question, are these things actually matched up into talking pairs? Here, Ted's work has really taken that to the next level and has actually explored specific signaling pairs and potential communication paths. When we start to understand what this actually means from the potential combinations and permutations of molecular switches that can lead to the presentation of pain, it means in the worst case, we're dealing with 2 to the power of 332 options, and if I'm super conservative, it's roughly 2 to the power of 80 options to signal varieties of signals that can create pain potential signature. What this means is that there are more potential molecular paths just at the bio level to generate pain than there are stars in the universe. Now, I want to re-emphasize that. There are more potential molecular paths to pain at the nanoscale than there are stars in the universe. Now, just also remember, this is just at the level of the nanoscale. It's only at the receptor-ligand pairs. It's only the proteins. It's only the dorsal root ganglia. It's only a binary yes-no, not a graded response. And it is only at the glial to neuron interactions. That means that literally what you saw there was the scientists' minds blowing, because that's how complicated a challenge we are currently facing to understand pain. So pain is complicated. It is extremely complicated to understand in the human setting when we can ask the situation, when we can ask and speak a language common to the pain experience. We are now acknowledging that it is so complicated that it spans across multiple systems, and there are so many different variations and combinations and permutations to pain that no wonder it is so prevalent throughout a wide variety of current years lived with disability. So is all hope lost? No. In fact, it creates an immediate need for us to focus on this attention to this challenge of pain through a different approach. And so I want to unpack with you this concept of convergent science. So what is convergence? Convergence is not new. There is an entire academic literature around the importance of convergence principles in creating and creating teams of disparate capabilities to tackle very large challenges. Now, what's important about the creation of these convergent teams is that there needs to be the correct culture. You need to have diverse people who have diverse educational capabilities, who are willing to partner together in long-term relationships, supported by good long-term funding to enable these outcomes. Now, the best example that I can give you of a convergent team is that embodied within the Big Bang Theory. What you have here are diverse teams of physicists and neurobiologists, in the case of Sheldon and Amy, who are undertaking more than just transactional collaborations in the laboratory. They're actually sitting down and working through and nutting out the challenges. And I can see the approach that TAWC takes in addressing the challenges that we are facing in animal welfare is this embodied absolutely in taking the conversations to more than a transactional collaboration, to a relationship level. So it is absolutely critical that we start to explore things from different environments to different levels in a collaboration framework. So that means that a wet lab individual needs to go into the theoretical world and have conversations, and the theorists or the dry lab experimentalists need to come into the wet labs to understand what the challenges are, so we can actually have more than this transaction to actually lead to relationships. Now, what is key to all of this is a development of trust. And I think we can see within the Animal Welfare Collaborative that trust has been central to so many of the strides forward that we've gained because there has been a mutual understanding and a mutual respect which has developed into a trusting relationship. Now, this means that when we start thinking about building our new workforce capability, the old training model of having a generalist understanding and only one thing you are good at is not sufficient for the future convergent science programs. We need to be able to create students and retrain our existing workforce of T-shaped capabilities into this comb shape of diverse depths of expertise across wide-ranging activities. And that's where key elements of conversation, collaboration, and relationship and trust come in as depths of expertise in other domains. So how do we actually go about doing this? How can we advance this field of pain, the Animal Welfare Collaborative, further? Well, I want to talk to you about this concept of bench to boardroom and really taking ideas from a lab bench experience, not to have the bookshelf as the destination for academic activities, but to actually translate and extend these through to adoption to have impact. Now, a key part of this must be that we have to acknowledge that this has to be consumer-centered. That is, we can't be doing things about something without that community involved. And this is, again, a long-held, long-understood model, which actually has arisen out of the disabilities advocacy, that we do nothing about us without us. Now, for pain consumers, those who suffer pain out in our community, it is critical that we actually engage in the design and research development phases with those pain consumers in mind. Similarly, as we look to tackling the challenges of mitigating pain in animals, there is no point a bunch of academics talking about solutions without having the diverse range of stakeholder groups at the table as we start to have these conversations. Again, a central activity of the TAWC Talks. We also need to acknowledge, and this is critical for pain, that statistical significance is not sufficient. I know that there are statistically significant treatments that we can administer for acute hyperacute pain. Those are opioids. Opioids are significantly and statistically significant efficacious agents to create pain relief in patients. However, opioids in the hyperacute setting still only work in roughly 1 out of every 2 to 1 out of every 4 people upon the first dose. That means when I say work, it's not a statistical significant work, it's a patient-centric significance of reduction in the amount of pain. So if we think about the scale of 1 to 10 of pain relief, to receive an opioid you need to be scoring greater than 4, so you might be at a 5 or a 6, and your pain reduces down to a 4. That does not mean you don't have pain anymore, it just means that statistically your pain has been reduced. Surely we need to evolve beyond just simply, yay, we reached statistically significant reductions in pain. Rather, we need mitigation strategies that create a pain relief that is sufficient to be community acceptable, society acceptable, and industry acceptable amount of pain reduction that leads to the benefit for, in the case of animals, the animal welfare, and for humanity, for humans, a reduction in pain that is meaningful to the individual and their family and community. Part of this journey, and specifically in the Australian context, is that when we start looking at academic research around the funding of that, we can't be thinking of these grants as funding to then go and do whatever we want with, but rather we need to be acknowledging that these are actually investments in the future with expected returns. Now, this is clearly the case and has been the last government's and the current government's messages that research funding must be tied to impact. That is not to say that fundamental science should not be invested in. Rather, it means that we should be doubling down on fundamental science that is embedded within programs of work that allows for the identification of promising new opportunities from that fundamental science and then enabling the returns of impact through the utilization of that science. Again, ensuring that these are consumer-centered, end-user, stakeholder-centered returns on investment in forms that are practical. Now, for human pain medicine in Australia, there has been a national strategic action plan for pain management. As you will have seen, human pain is a massive problem. We know that animal pain is a massive problem. We have not done anything yet in human pain that actually works, but we do have a national strategic action plan for pain management. And 6.1 of this National Action Plan stipulated the establishment of a new National Institute for Pain Research. Now, I've been part of a steering committee that has started to explore what this actually might look at, and I'm fortunate enough to chair that steering committee. And so where we have evolved to is that we don't want an institute, a bricks and mortar activity that solely serves one geographic area or one particular discipline, but rather we need to create an alliance that generates pain solutions and research that drives pain solutions across a broad breadth of areas of research in both human and animal applications. Critically, that allows us to take concepts and ideas of potential impact, solution to a true impact stage, that is connecting fundamental science to extension and adoption. So the Australian Pain Solutions Alliance, or the Australian Pain Solutions Research Alliance, seeks to create a capability based on the convergence science principles of culture, people, education, partnering with enduring funding. And it's been exciting to see the Animal Welfare Collaborative, veterinary groups, right through to regulating bodies, AVPMA, being at the table and influencing how this alliance is being established. The vision is for the human element of this, and I think it would be not unrealistic of us to actually implement the animal version as well. And let me start by stating the human version. All Australians shall live unburdened by pain. And I think it does actually therefore work. All animals in Australia shall live unburdened by pain. And the key part here is to unite consumers, researchers, health professionals, industry and government to propel the discovery and delivery of effective pain solutions. And we can add here to humans and to animals. So now I'd like to wrap up, but I'd like to wrap up in a slightly different way for this particular point. We are going to be a multi-planet species by 2032. By the time Australia hosts the next Olympic Games that will be in Queensland 2032, we will be on Mars. That is only a decade away. NASA's already planning for it. It probably won't be a government agency that gets us there. It will probably be the private sector that actually takes us there. By 2032, we will have undertaken the next layer of humanity's progression to become a multi-planet species. My challenge to you is, will we and the animals in our care still be burdened by pain? I hope that through taking a convergent science approach and adopting principles that TAWC absolutely is embodying, we will have tackled this challenge by rapidly accelerating the translation and adoption of exciting new pain measurement and mitigation solutions. And so whilst this is just the first part of my talk, series of talks, I do want to emphasize that this does take everybody leaning against the same wheel, working collaboratively in a trusting partnership across multiple sectors, working towards a common goal. Thank you.